Hello, we, Rebeca Pérez Gavilán, Karen Ambriz, Valeria Martinez, Nicole Moreno, and Karen Esquivel, come from UVM Campus Cumbres, and along our teacher, Susana Castillo, we were assigned on our chemistry class to talk about the nucleic acids. What are nucleic acids? They are molecules that allow organisms to transfer genetic information from one generation to the next. There are two types of nucleic acids: deoxyribonucleic acids, which are better known as DNA, and ribonucleic acid, also called RNA. Now, what is DNA and RNA? DNA is a nucleic acid that contains the genetic instructions used in the development and functioning of all modern living organisms that have a blueprint function of biological guidelines that a living organism must follow to exist and remain functional. A medium of long-term, stable storage and transmission of genetic information, and a double-stranded structure that has two nucleotide strands, which consist of its phosphate group, 5-carbon sugar and 4 nitrogen containing. The information found in DNA determines which traits are to be created, activated, or deactivated. While the various forms of RNA do the work, it helps carry out DNA's blueprint guidelines and transfer genetic code needed for the creation of proteins from the nucleus to the ribosome. Its structure is single-stranded. Like DNA, RNA is composed of its phosphate group, 5-carbon sugar, and 4 nitrogen containing. A nucleotide is the basic structural unit and building block for DNA. These building blocks are hooked together to form a chain of DNA. It is composed of 3 parts: 5-sided sugar, phosphate group, and nitrogenous base. The sugar and phosphate group make up a backbone for the DNA double helix, while the bases are located in the middle. A chemical bond between the phosphate group of one nucleotide and the sugar of a neighboring nucleotide holds the backbone together, and chemical bonds between the bases that are across from one another hold the 2 strands of the double helix together. Then we have the nitrogenous bases. We have 4 types of bases in DNA, which are called adenine, cytosine, guanine, and thymine. Bases are part of DNA that stores information and gives DNA the ability to encode phenotype. A person's visible traits, adenine and guanine, are purine bases. These are structures composed of a 5-sided and 6-sided ring. Cytosine and thymine are pyrimidines, which are structures composed of a single 6-sided ring. Adenine always binds to thymine, while cytosine and guanine always bind to one another. This relationship is called complementary base pairing. These complementary bases are bonded together via hydrogen bonds, which can be easily broken apart when the DNA needs to unzip and duplicate itself. The popularly recognized DNA form is due to the chain that is formed between all the bases. Since the number of bonds the cytosine and guanine have is 3 and the thymine and adenine is 2, the form gets twisted and leaves it as we all recognize it today. For sugar, we have the 5-carbon sugars, ribose and deoxyribose, which are important components of nucleotides and are found in RNA and DNA respectively. The sugar is found in nucleic acids are pentose sugars. A pentose sugar has 5 carbon atoms. A combination of a base and a sugar is called a nucleoside. Ribose, found in RNA, is a normal sugar, with 1 oxygen atom attached to each carbon atom. Deoxyribose, found in DNA, is a modified sugar, lacking 1 oxygen atom. This difference of 1 oxygen atom is important for the enzymes that recognize DNA and RNA, because it allows these 2 molecules to be easily distinguished inside organisms. And finally, we have phosphate. Its chemical formula is PO43, and it is a chemical compound. It is made up of 1 phosphorus and 4 oxygen atoms. When it is attached to a molecule containing carbon, it is called a phosphate group. It is found in the genetic material DNA and RNA, and is also in molecules such as adenosine triphosphate that provides energy to cells. Phosphates can form phospholipids, which make up the cell membrane. Phosphate is also an important resource in ecosystems, especially in freshwater environments. are activating proteins. Phosphate groups are important in activating proteins so that the proteins can perform particular functions in cells. Proteins are activated through phosphorylation, which is the addition of a phosphate group. Protein phosphorylation occurs in all forms of life. Dephosphorylation, the removal of a phosphate group, deactivates protein. And remember, just as Mehmet Oz said, your genetics load the gun, your lifestyle pulls the trigger.